Hello everyone, welcome back to Pathomed. Today let us study about gastrointestinal stromal tumor. So just this topic is important for both theory and practicals of MBBS and MD DNB examinations. So let us begin with introduction and all. So this is also given as a common spotter chart in the practical vivas as well. So this chart shows you a huge mass and it says to arise from the gastric wall surface. They ask to give your diagnosis and the cell of origin. So let us begin the topic. So gastrointestinal stromal tumor. As the name suggests, it is a stromal tumor, means it is a mesenchymal tumor. Stroma is mesenchymal tissue. So it is the most common mesenchymal tissue, mesenchymal tumor of the GIT, gastrointestinal tract. So the size can vary from as small as less than 2 centimeters to as large as more than or equal to 30 centimeters. As shown in the image, this is a very huge mass said to arise from the gastric wall surface. The histology part will come back to it once I discuss about the histological variants. So, so the cell of origin is gastrointestinal tumor, gastrointestinal stromal tumor. It is said to arise from interstitial cells of Cajal, interstitial cells of Cajal, or you can pronounce it as Kajal, Kajal or Cajal. These are nothing but the pacemaker cells. These are the pacemaker cells of the GIT. They bring about gut motility and the peristalsis. Pacemakers bring about motility. It brings about action potential. The pacemakers of the GIT is called interstitial cells of Cajal. These cells are responsible for gastrointestinal tumor, stromal tumor. So I will discuss about the anatomy here on histology. For example, in the gut wall, this is the epithelial layer from inside to outside I'm coming. First layer is the mucosa. This is the mucosa lined by this epithelium, columnar epithelium. Beneath the mucosa is the submucosal layer. Submucosal layer. It is composed of loose fibrous tissue. Beneath the submucosa is the muscular layer. Inner circular Inner circular, outer longitudinal layer. Outermost layer is the serosa. Serosa contains the fibrous tissue with blood vessels and etc. Everything. So, mucosa, beneath the mucosa is the submucosa, comes the muscle layer and the serosal layer. The pacemaker cells, that is interstitial cells of Cajal, are present in between the muscle bundle fibers. So in between the inner circular outer longitudinal, these pacemaker cells are present. These are the interstitial cells of Kajal. So because of certain mutations, the activity of Kajal cells become increased activity, increased cell proliferation. They proliferate in uncontrollable way. Uncontrolled proliferation eventually leads to cancer, that is tumor. So hence ultimately it leads to GIST. So this is all about the histology part. I have told you the interstitial cells of Cajal arises from the interstitial cells of Cajal. The most common sites being, coming to the sites, most common sites being the stomach. So more than like 50%, 50 to 60% arise from the stomach wall, followed by small intestine, followed by colon, rectum, followed by least is the esophagus. Esophagus is the least site, less than 5%. So stomach being the most common site. I've told you about the mutations. Certain mutations lead to GIST. What are those mutations? First, the most common mutation being the secret gene. Secret is a gene. It has like intrinsic tyrosine kinase activity. It is a tyrosine kinase receptor. Tyrosine kinase receptor means it has that autophosphorylic activity. So whenever this receptor gets activated, this uncontrolled cell proliferation. So c-KIT gene, which is a tyrosine kinase receptor, When this gene gets mutated, it leads to uncontrolled cell proliferation of the interstitial cells of Cajal, ultimately leading to GIST. This is the most common gene, like c-KIT gene responsible for 85% of the GIST. The second gene being, wait a second, the second gene being PDGFRA gene, almost 15, like less than 50%. It stands for platelet-derived growth factor receptor alpha. Platelet-derived growth factor receptor alpha. It stands for platelet-derived growth factor receptor alpha. The third variant, that is third mutation being, that SDH deficient GIST. SDH means succinate dehydrogenase. Succinate dehydrogenase enzyme deficient. This is the most common variant seen in pediatric age group. It is a pediatric GIST. It is a pediatric GIST. So, I would like to discuss the histological variants. Before proceeding, so there are three variants. One is the spindle cell variant. Spindle cell variant is most commonly seen with c-KIT mutation and PDGFR A mutation. Next is the epithelioid GIST, seen most commonly with SDH deficient GIST, SDH deficient GIST, that is the pediatric GIST, pediatric, sorry, pediatric GIST. Third is the mixed variant. Mixed means it has both spindle cell and epithelioid cells, combination of both types. So the c-KIT mutation shows you spindle cell, platelet-derived growth factor receptor alpha mutation. It most like usually it shows you spindle cell type. SDH, the pediatric one, shows you the epithelioid variant. Let us begin the histological types. This slide shows you like spindle cells. Most of the pictures spindle cells. Spindle means they are elongated with tapered ends. These are elongated cells. You can visualize the nuclei of the cells. These are the purple stain are the nuclei. They are elongated, they have tapering ends. Like tapering means they are spindle. The ends are pointed. This is a spindle cell. This is said to be a spindle shape. So this is a spindle cells. So smooth muscles they show you spindle cell. So and also they are arranged in like fascicles. They show you fascicles. Fascicles mean the all the smooth muscle, the nuclei they're pointing in one direction. They have one direction, unidirectional. This has one direction, see. This has one direction. This has one direction like this. So it has a certain direction. These are called fascicles. They show fascicles of smooth muscle cells. It is a spindle cell variant. Most, like 100%, the slide shows you spindle cells. Spindle cells. Elongated are the spindle cells. They are arranged in fascicles, unidirectional. This is the first variant, that is the spindle cell type. Coming to the second, that is epithelioid. Epithelioid means round. They are round. Plump means swollen. Cytoplasm is eosinophilic. So you find this cell. This is one cell. They are round, like plump. It appears to be swollen, round, plump. Also, you notice vacuoles. There are like beside the cells there are vacuoles. Paranuclear vacuoles. These are the paranuclear vacuoles, common in epithelioid variant. Epithelioid means you visualize round plump cells with eosinophilic cytoplasm. Coming to the third variant, that is the mixed variant, you find both, like there are spindle cells, these are the spindle nuclei, spindle. Also, you find epithelioid, round plump cells. Both in one picture, epithelioid and spindle both in one picture is said to be mixed type. Also in this slide you find this like dark stained nuclei, extremely dark stained, extremely dark stained nuclei here also. These are nothing but mitotic figures. Mitotic figures means cell in a dividing state. When the cell is about to divide into two, the nuclei showing the changes of division is called a mitotic figure. Mitotic figures. So here this flower shaped, it is like prophase. This is also a prophase, like flower shaped nucleus. Here it is like about to like separate out, to separate. It is about like anaphase. Anaphase. So mitotic figures are also like a very important diagnostic clue for tumor, and it is like most commonly seen in GIST as well, and it helps in like categorizing its prognosis. Like, is it good prognosis or bad prognosis can be decided based on the number of mitotic figures you see. So I'm telling about this prognosis. The first prognostic marker is the mitotic figures. Mitotic figures that you have just seen. It is categorized as like less than 5 mitotic figures per 50 high power field. It is said to be like good. It is said to be good. Less the number of mitotic figures is good. Less number of mitotic figures is good. If it is more than or equal to 5 mitotic figures per 50 high power field, more number of mitotic figures means it is bad. Sad. It is bad. The second prognostic factor being the site. Like stomach, most common location is stomach. Stomach is a favorable, like it shows a good prognosis. Third prognostic factor is the size. Obviously, less the size, like less the size, less the size, good prognosis. Like less than 2 centimeters. The size is increasing. Increased size means bad prognosis. Increased size is bad prognosis. Coming to the IHC markers, immunohistochemistry. Each tissue expresses their own set of antigens. Since GIST has this because of c-KIT gene mutation, the marker for c-KIT is CD117. So the tissue expresses CD117 on the surface. So CD117 marker is shows you membrane positivity. Membrane positivity means there's like brown staining at the membranes. This brown staining. So brown means it is high HC is positive. Any high HC in the world, it shows you brown means positive. Blue, light blue. Here this you see light blue means negative. So we have done this. This is a CD117 IHC giving you like strong membrane positivity. It is a membrane positivity. And CD11 because since most of the GIST are because of c-KIT mutations, this is highly sensitive, but it can also be negative. That CD117 can also be negative in PDGFRA mutations because if the GIST has occurred because of this mutation or SDH deficient GIST. In these two variants, CD117 is negative. Then what? Then the second marker, DOG1. Dog one. Dog one is given, it is named after GIST only. It is like full form is diagnostic. Diagnostic of GIST. Diagnostic of GIST. So it is especially useful when CD117 is negative and you are suspicious for GIST. So it shows you both like diffuse membrane and cytoplasmic positivity. And also like CD117 is it is highly sensitive but it is not specific because CD117 can also be positive apart from the GIST. It can also be positive in AML, it can be positive in melanoma, it can be positive in mastocytosis and seminoma. So these five conditions show you positivity for CD117. Hence, it is non-specific. It is not specific. It is sensitive for GIST, but not specific. In such cases, also you can do DOG1. DOG1 is highly specific. It is highly specific. for GIST. So the differential diagnosis, sorry. So the differential diagnosis for GIST are they are very similar, like histologically they are very similar to leiomyoma and leiomyosarcoma. Both are smooth muscle tumors. One is benign, one is malignant. But the difference on histological features are like leiomyoma also it is also a muscle tumor, smooth muscle tumor, but the nuclei here are cigar shaped. They have blunt ends. They have blunt ends. Whereas in GIST, it has tapered ends like this spindle cell. So apart from the histological differentiation, based on IHC also, like leiomyoma, leiomyosarcoma, they show positivity for SMA, that is smooth muscle actin and desmin. Both are muscle markers. Smooth muscle actin, desmin are positive for these both. Schwannoma, again, it is positive, shows highly strong positivity. For S100, it shows strong positivity for S100 schwannoma. Because the schwannoma also shows you like the spindle cell kind of thing. So to differentiate schwannoma from just you can do this S100. And also it is similar to metastatic melanoma, where melanoma is positive for both S100 and HMB45. These are specific IHC markers for metastatic melanoma. All the IHC markers in the world, they like to interpret brown means positive, light blue means negative. Whichever marker you take, they show these two colors only. You have to be like, it is like the markers are different. You are doing staining for this P63, doing staining for P53, doing staining for Desmond, doing staining for SOX10, S100, HMB45. The names are different, but on the slide they show you blue and brown stains. So let us summarize. Let us summarize. So gastrointestinal stromal tumor. It is the most common mesenchymal. Stroma means mesenchymal, so it is a mesenchymal tumor of GIT. Size ranges from small to very large, and in this you can see that two population. There are epithelioid cells. These are round cells also seen. Spindle cells are also seen. So I say it as a mixed type of variant. Cell of origin is from interstitial cells of Cajal, which are the pacemaker cells. Most common site being the stomach. Mutation most common being the c-KIT mutation, which is a tyrosine kinase receptor. Mutation of it leads to uncontrolled cell growth. The second most common mutation being the platelet-derived growth factor receptor alpha. In pediatric age group, the succinate dehydrogenase enzyme like SDH deficient just is common. Variants are spindle cell variants, spindle with elongated tapered ends. Epithelioid means round, plump, mixed to show these both features. So this is the fascicles of spindle cells. You see fascicles of spindle cells. These are the round plump cells, epithelioid. This is the mixed cell. This is the mixed kind of both spindle and round plump cells. Also, you see mitotic figures. Mitotic figures are important for categorizing the prognosis. Less than 5 means good, more than 5 means bad. Side stomach is a favorable, it has a good prognosis. Size, lesser the size, low size means good prognosis. Increased size means bad prognosis. And IHC markers, most of the mutations are because of c-KIT. c-KIT marker is CD117. CD117 shows you diffuse membrane positivity. It is highly sensitive but not specific because apart from just it is positive in these conditions also. And also in other conditions where CD117 is negative is PDGFR-A mutation and SDH deficient just in those the CD117 is negative. In such situations, dog one is like highly specific, it comes to help. Diagnostic of GIST is highly specific for GIST. It shows you both membrane and cytoplasmic positivity. Differential diagnoses are they are very similar to leiomyoma, leiomyosarcoma, schwannoma, metastatic melanoma. To differentiate, you do IHC markers. Leiomyoma, leiomyosarcoma positive for SMA and Desmin. Schwannoma is highly positive for S100. Melanoma for S100 HMB45. So this is all about just thank you everyone. Please Like, if you find this video useful, please like, share, and subscribe to my channel. Thank you, everyone.